Hey everyone, my name is Madison, and today I'm going to introduce my protein, which is the MCR1 protein, also known as the melanocortin 1 receptor. This protein is a G protein coupled receptor, and it plays a vital role in pigmentation by regulating the production of melanin, which is the pigment responsible for color in your skin, hair, and fur, in this case for mice. MCR1 is found in the cell membrane of melanocytes, which are the cells that produce the melanin. And we're going to focus on two different types of melanin: eumelanin, which is responsible for producing darker pigment, and pheomelanin, which is responsible for producing light pigment. Variations between these two different types of melanin is how we see coloration across all different types of species. So when MCR1 is functioning normally, it signals melanocytes to produce eumelanin. So we naturally see a darker pigmentation occurring. However, when we have mutations in this MCR1, we can see alterations in signaling that is kind of going to shift the balance toward producing more pheomelanin, or seeing a lighter pigmentation produced. So these mutations contribute to all the differences we see in pigmentations across populations and species. So for purposes of this project, we're going to focus on the beach mice, where this specific amino acid, very small amino acid change, results in a lighter fur coloration for these mice who have naturally darker fur. It does seem very small, but it does have a huge impact, especially on the evolutionary scale, allowing these mice to blend in with their sandy environment and reducing their chances of being seen by the predators and increasing their rate of survival. So with that being said, in this video, I'm going to break down MCR1 structure and demonstrate how a single amino acid mutation at position 67, where arginine is replaced by cysteine, affects its function and shape. So here I've represented primary structure using these beads on this string. This overall represents the amino acid sequences. To note, all the amino acids are definitely different, but I highlighted here, this is the normal strand or amino acid chain, and this is the mutated one. This yellow is showing the mutation of the cysteine at position 67, where arginine would originally be. Here's the secondary structure of the MCR1 protein. This is showing the alpha helices that the primary structure comes together to form. Here we can see that the regular formation of the secondary structure is tightly bound and looks pretty normal. And then here is the mutated structure with the cysteine position at 67 amino acid. We can just see from this that the interactions that these two show is that these are tightly interacting together, because arginine is a positively charged amino acid and therefore engages in strong ionic interactions with negatively charged molecules, and overall makes the protein highly hydrophilic. Cysteine, on the other hand, is not positively charged and is neutral. So therefore, these interactions are not as tight as this shows here. Cysteine has a thio group, which is a sulfur hydrogen group that allows it to participate in disulfide formation with other cysteines. So that can be another factor to why we see such great conformational changes in the secondary structure of the protein. So here is the tertiary structure I've chosen to represent the MCR1 protein. Again, this protein is a G protein coupled receptor, so it has a complex three-dimensional shape that is crucial for its function. We can see here it consists of these 7 alpha helices that span the cell membrane, and they are weaving in and out of the lipid bilayer. This lipid bilayer is shown as this box. We can just say, for purposes, that this side is the extracellular space and this side is the cytoplasm. We can see here that these helices are embedded in the hydrophobic environment of the membrane, overall helping the protein stay in place while it's interacting with the signaling molecules that are on the inside and outside of the cell. So here we have these extracellular loops of MCR1, which extend to the extracellular matrix and are responsible for binding signaling molecules like the alpha melanocyte stimulating hormone. which is known as alpha MSH. And this interaction triggers a cascade of intracellular events that regulate the pigment production. These intracellular loops extend in the cytoplasm and play a role in transmitting the signal by interacting with G proteins. Once activated, the MCR1 stimulates the production of cyclic AMP, which ultimately leads to increased eumelanin, or the darker pigment, production in melanocytes. Here I'm showing the mutation in position 67 with the replacement of arginine with cysteine. It's in one of the transmembrane helices right here, and it's in the first intracellular loop. So overall, arginine is positively charged and cysteine is neutral and contains a sulfur group. So the substitution can really affect the folding and stability of the protein. It's kind of hard to show, but there is a little difference here with this yellow marker. This small change really does affect how the helices interact, and it weakens the receptor's ability to signal properly. So this is why we see in beach mice with this mutation, they have a decrease in this eumelanin production and overall have lighter fur, because these signals that we're receiving from the outside can't be relayed to the inside to overall produce this signaling cascade that produces more eumelanin to further produce genes that express the darker pigmentation.